gonna share my wondrous experience in DRILS. Yeah, DRILS is the best platform for the young scientists to accelerate their thoughts towards science. We visited DRILS to know more about the zebrafishes because they are gonna be the next model organisms, as 70% of its genome matches with the human genome. We learn a lot about these fishes there, and even we came to know that they can even regenerate the damaged heart tissues, and scientists are very curious to implement this to humans too. We even micro-injected the embryos of these fishes, and it's a great experience for us. As these fishes have transparent embryonic stages, it's very easy to observe its organ functioning, and by anesthetizing the fishes, we observe the blood circulation, heartbeat, gill functioning, etc., which is a delightful experience for us. Scientists explained us how it is implemented in drug efficacy and how they have succeeded in sorting out many neurological problems by taking zebrafish as the model organism. It's an immense opportunity for us to visit DRILS, and we gain a lot of knowledge, and this shown us a new path towards the research and eagerness in the subject and increase the way of thinking. Thank you.
As undergraduates, this is our first ever exposure to the research field and laboratories. We were shown the different kinds of equipments and the instruments that are used in this laboratory and explained about their working in brief. The main focus of the day was on zebrafish that is currently being used as model organism in research, and we were taught different things about these fishes, like the differences between the males and females, their breeding, feeding, maintenance, and how they're being used to test the drug efficacy, and many more aspects. Visualizing the different staged embryos, their organs and blood circulation under the microscope was very interesting. Learning how to micro-inject a single cell staged embryo was the greatest of all the experiences that we had on that day, and I really feel very lucky and privileged to get such a practical experience at my undergraduate level itself. Knowing about many of their previous publications and their current research in the laboratory created more enthusiasm in me and created interest in me to learn about it in detail. This visit to DRILS helped me understand that keeping myself updated with the current research and current research publications will definitely increase my interest and enthusiasm towards the subject and keep my motivational levels high. Finally, we really had a very great and enriching learning experience, and it has filled me with lots of motivation and inspiration.
Was meticulous. It provided us opportunity to learn about the various aspects of research, and the main focus was on zebrafish, like culturing the zebrafish, the optimal conditions needed for its growth and feeding. The micro-injection part was hard, yet it was very interesting. Visualizing the blood circulation was one of the best experiences we had, and we have gone through the many articles published by them on zebrafish, and they included the neurological disorders and the usage of nanoparticles. Altogether, it was the best experience we ever had, and it increased the impulse towards research.
Hello everyone, today I am here to share about visiting Dr.Reddy Institute of Life Sciences. Visiting DRILS was one of the memorable days in my life. At DRILS we were taught about how zebrafish is used as model organism and how to differentiate between male and female zebrafishes, and also about how to culture the zebrafish. The best thing at DRILS we did was micro-injecting. Micro-injecting into single cell stage embryos was very interesting, and we were very excited to do that. Studying micro-injection theoretically was very different thing when we do it practically. And they also explained about the research articles they publish in which zebrafish is used in every scenario. Undergraduate students like us should visit research field so that they will know more about the research field, and they will enhance their knowledge towards research.
Visiting DRILS was one of the greatest opportunities that we had at our UG level. We were interested to visit DRILS to know about zebrafish. Culturing and breeding of zebrafish stands the main focus, as they make the main part to deal with them. Their maintenance, feeding, drug controlling aspects dealing with neurological disorders were interesting to know. The best part of the visit was about microinjection, which was one of the most interesting and difficult aspects of research. I feel lucky and privileged to perform it. The articles on zebrafish helped me to know current research aspects. At this visit, I realized the fact that science is not about reading books, it's about exploring things. I extend my heartfelt thanks to Mr. Raghavendra sir, a PhD student at Zebrafish Facility, Dr. Shweta Singh ma'am, Scientific Research Officer, and Dr. Kinran Shetty sir, Principal Research Scientist Head at DRILS, for this wonderful experience.